I was starting making a PhD in oncology, and when I was making my experiments, I was quite disappointed by the limitation of the in vitro models that I was using. And so I decided to create something much more sophisticated with a multi-tissue system.
Today, drug discovery is inefficient. We spend more than $2 billion per drug and more than 10 years of research and development. But why? Because we use in vitro methods and animal models that are poorly predictive and poorly relevant to reflect the human biology. But now, we are assisting to a revolution with new technologies that are coming, like 3D cell culture methods to screen a huge amount of compounds, and then we assess the drugs with a more sophisticated methods called the human on a chip, where we put several organs on the chip to mimic the organization of the human body. But at FluoSphera, we identify a technology gap in between, where we need the relevance of the human on a chip, but with the flexibility and the throughput of 3D cell culture methods. So, at FluoSphera, we develop a solution using the power of fluorescent light to mimic the organization of the human body. Briefly, we isolate tissues from the human body, like the heart and the liver together, into fluorescent capsules for the recognition of the tissues. Like that, we create liquid microphysiological systems. And then we use fluorescent probes to track the activity of drugs on these communicating tissues.
So here we have the liver, here we have the brain, and then we can combine them together to make a liver-brain system. We are in a very active space today because organoids, spheroids, this is really active topics today. And we are really different from these competitors because we combine different tissues together. When we speak about organoids, this is a single type of organoids. We almost never combine the different types together. So this is a way that we are different and that we are better to predict than the effect of drugs on these kind of systems.
We proved already that our technology is working on tamoxifen, a famous drug used against breast cancer that was saving millions of lives. But tamoxifen was discovered by accident because it is not efficient at all in vitro. And why? Because when we test tamoxifen in vitro, we use the breast tissue alone. And tamoxifen needs to be activated by the liver first. And with our technology, we can combine the liver and the breast together, and we can capture the efficacy of tamoxifen, meaning that we can also discover other drugs like tamoxifen that will save millions of lives. So we target the step of lead optimization. This is a specific step of drug discovery, one of the most important ones, but also the most expensive. First, our solution can be used much earlier compared to chip-based MPS. Thanks to that, we miss less promising compounds, and so we can discover potent drugs for the patients. Then our technology is very easy to use, so it decreases the barrier of adoption within the market, and it is highly flexible, so we can develop systems for most pharmaceutical applications. Please join us to revolutionize drug discovery with FluoSphera. Thank you very much.